Welcome, curious minds, to the deep dive. I'm really excited to kick things off today with this image that just seems completely impossible. Picture this, the massive empty stretches of the Sahara Desert, and then right there, buried deep in the sand whale bones. Huge marine creatures. How on earth did they end up in one of the driest places on the planet?
It's really something, isn't it? It's not a trick, though.
No, not at all. It's actually this incredible clue about our planet's past, how dynamic it is, and, well, the amazing story of how life transforms.
Yeah, the Sahara whale fossils. They're not just weirdly placed bones, they're solid proof of huge changes over immense timescales. They actually show how early whales moved from land back into the sea.
And then the land itself changed around them. Wow.
Exactly. And that leads us right into our main topic for this deep dive, evolution.
Right, and we should stress, this isn't just a history lesson. Evolution is the absolute foundation for understanding why life is the way it is today.
It explains the unity, like how nearly everything shares the same basic genetic code.
And the incredible diversity from those ancient whales to bacteria.
And all the intricate adaptations you see, how every organism is sort of tailored to its environment.
So our goal today is to take a fantastic biology text, Campbell Biology in Focus, and really pull out the crucial ideas about evolution.
We'll walk you through the big concepts, the discoveries that changed everything, and the processes that are still happening.
We'll show how tiny changes, like at the genetic level, can lead to these massive outcomes over time.
Think of it as your essential guide to understanding the core of life itself.
Okay, let's dive into life's endless forms most beautiful, as Darwin put it. We'll start with him, but first, maybe touch on what came before. For a long time, people thought species were just fixed, unchanging.
That was the prevailing view, absolutely. Linnaeus gave us our classification system, genus, family, order grouping similar species, but he saw it as revealing a divine pattern, not shared ancestry.
But the ground was already shifting a bit, wasn't it, geologically speaking?
Yes, exactly. Geologists like Hutton and Lyell were talking about slow, gradual changes shaping the Earth. Rivers carving canyons, mountains rising over millions of years.
Which suggested Earth was incredibly old, much older than people thought.
And that huge timescale was crucial. It opened up the possibility that life, too, could change gradually over long periods.
Then there was Lamarck. He had this, well, partly right idea.
He did. He proposed that organisms adapt to their environments and that gradual change and adaptation were key drivers. He saw that link between life and its surroundings.
Even though his mechanism inheriting acquired traits, like giraffes stretching their necks and passing that on, wasn't quite right.
Right, that part was incorrect. But even a wrong hypothesis can be valuable if it pushes thinking forward, asks the right kind of questions. Lamarck really focused attention on adaptation.
Okay, so enter Darwin. His voyage on the HMS Beagle, that 5-year journey, seems like where it all started to click for him.
It was fundamental. Charting the South American coast, collecting fossils, observing everything, he noticed patterns.
Like how temperate species in South America look more like tropical South American species than, say, European temperate species.
Precisely. And the fossils he found often resembled the living organisms in that same region. Little puzzles.
And the Galapagos Islands. That was the real turning point.
Oh, absolutely. The finches, especially. Each island seemed to have its own unique finches, but they were all clearly related. The main difference was their beaks.
Adapted for different foods, right? Like seeds, insects?
Exactly. He started thinking they must have come from the mainland, and then diversified, adapting to each specific island. This led him to formulate the idea of adaptation inherited characteristics that boost survival and reproduction in a particular environment.
And the mechanism driving this adaptation. That's the big one, natural selection.
Yes, the idea is that individuals with certain inherited traits are more likely to survive and reproduce than others because they have those traits.
It helps to think about artificial selection, maybe? How humans do this.
That's a perfect analogy Darwin himself used. Look at dogs all descended from wolves. But we've selected for traits we like, creating breeds from Chihuahuas to Great Danes.
Or even vegetables, like how broccoli, cabbage, kale, they all came from selectively breeding the wild mustard plant. We picked the traits.
Exactly. We're the selecting agent there. In natural selection, the environment does the selecting.
So Darwin built this logic. What were his core observations?
Two main ones. First, variation. Individuals within a population vary in their inherited traits. Think about ladybugs, different numbers of spots, slightly different colors. There's natural variation.
Okay, variation exists. What's the second observation?
Overproduction. More offspring are produced than can possibly survive. A maple tree drops thousands of seeds, a frog lays hundreds of eggs. Most won't make it.
So variation plus overproduction leads to.
Two key inferences. First, differential survival and reproduction. Because of the struggle for existence, individuals with traits that give them an edge in their environment are more likely to survive and leave more offspring.
Makes sense. Better camouflage, faster speed, resistance to a disease.
Whatever helps in that specific context. And the second inference follows directly. Accumulation of favorable traits. Over generations, those advantageous traits become more common in the population.
And this is critical. It's the population that evolves, its genetic makeup changes, not the individual changing during its lifetime.
Precisely. An individual bird's beak doesn't get longer because it needs to. Rather, birds born with slightly longer beaks might do better in a certain environment, reproduce more, and so the average beak length in the next generation increases.
The evidence for this whole process is, well, everywhere once you know what to look for. You mentioned seeing evolution in action.
Yes, we can observe it directly. Take soapberry bugs in Florida. In the south, where they feed on balloon vines with deep seeds, they have long beaks.
Okay.
But in central Florida, a new plant, the golden rain tree, with seeds closer to the surface, was introduced. Within about 35 years, the soapberry bugs in that area evolved shorter beaks. That's rapid adaptation.
Wow, 35 years isn't long in evolutionary terms.
Not at all. And think about drug-resistant bacteria, like MRSA. Penicillin arrives, works great for a bit, then resistant Staphylococcus aureus appears. Methicillin is developed, and boom, resistance within a couple of years.
How does that happen so fast?
Bacteria reproduce incredibly quickly. A generation can be just 20 minutes, so any random mutation that confers resistance gets selected for very strongly when the antibiotic is present. The resistant ones survive, multiply, and soon dominate the population. It's a huge public health issue driven by natural selection.
That really brings it home. So we see it happening now, but how do we trace these connections back through deep time? What about shared ancestry?
That's where homology is so powerful. It's about similarities resulting from common ancestry. Think about the forelimbs of mammals.
Like a human arm, a cat's leg, a whale's flipper, a bat's wing.
Exactly. They're used for totally different things, grasping, walking, swimming, flying. But if you look at the bone structure,
It's the same basic pattern. One upper arm bone, two forearm bones, wrist bones, finger bones.
right, it's the same underlying architecture, modified for different functions. It strongly suggests they all inherited that pattern from a common ancestor. Evolution tinkers, it remodels what's already there.
And this homology isn't just in bones, right?
Not at all. We see embryological homologies. For instance, early vertebrate embryos all have structures like a postanal tail and pharyngeal arches. Even if they develop into very different things, gills in fish, parts of the ear and throat in humans.
Remnants of our shared past.
And at the deepest level, molecular homologies. The genetic code itself is essentially universal. The DNA language is the same in you, in a mushroom, in bacteria. It's compelling evidence for a single origin of life.
Okay, so that's different from analogy, where things look similar but evolved independently.
Correct. Analogy or convergent evolution is when similar environmental pressures lead to similar adaptations in unrelated organisms. Think of the streamlined shape of a shark, a fish, and a dolphin, a mammal.
They both need to move efficiently through water, so they ended up with similar body shapes, but not because they inherited it from a recent common ancestor with that shape.
Exactly. Function dictates form independently. Homology is about shared history. Analogy is about shared environmental challenge.
So homologies point backwards. What about the direct storybook of life, the fossil record?
The fossil record is our time machine. It shows us that life was different in the past, that many species have gone extinct. Crucially, it documents evolutionary changes within lineages over time.
How do we know how old fossils are?
We use radiometric dating. By measuring the decay of radioactive isotopes in rocks around the fossils, or sometimes in the fossils themselves, we can get reliable age estimates. Carbon-14 works for younger fossils, up to maybe 75,000 years. For older stuff, we use isotopes with longer half-lives, like uranium-238 and volcanic layers.
And this allows us to see major transitions.
Yes, beautifully. The evolution of cetaceans, whales, and dolphins is a great example. Fossils like Pecacetus and Rhodocetus show intermediate forms between land mammals and fully aquatic whales, even showing changes in their ankle bones, linking them to hippos and other even-toed ungulates.
An incredible journey documented in stone.
Absolutely. The fossil record, combined with dating, gives us the geologic record, the sequence of life's history. From the earliest prokaryotes around 3.5 billion years ago, the rise of oxygen, the first eukaryotes, multicellular life, the Cambrian explosion of animal diversity, the colonization of land, it's all laid out.
And the continents themselves have been moving all this time. How does biogeography, the study of where species live, fit in?
It fits perfectly. Continental drift, driven by plate tectonics, has had a massive impact. Remember Pangaea, the supercontinent?
Around 250 million years ago.
Yes. When it formed, climates changed, coastlines vanished, extinctions happened. Then, as it broke apart, populations got isolated. This geographic separation is a major driver of speciation.
So related species might end up on different continents because the continents drifted apart after their common ancestor lived there.
Exactly. Or think about islands. Island species often closely resemble species on the nearest mainland because they likely descended from mainland colonizers who then adapted and diversified in isolation. Biogeography makes sense only in the light of evolution and Earth's history.
Okay, let's zoom back in a bit. You emphasize that populations evolve, not individuals. This is microevolution. Can you break down what allele frequencies changing really means?
Sure. So genes often come in different versions called alleles. Maybe a gene for flower color has a red allele and a white allele.
Got it.
The allele frequency is just how common each of those versions is in the population. If, say, 60% of the flower color alleles are red and 40% are white in one
And
generation,
in the next generation, it's 70% red and 30% white.
then the allele frequencies have changed. That is microevolution. Simple as that at its core.
What provides the variation for these changes? Where do new alleles come from?
The ultimate source is mutation or random changes in DNA. Gene duplication can also create new genetic material to work with. Then, for organisms that reproduce sexually, sexual reproduction is huge. It doesn't create new alleles, but it shuffles existing ones into new combinations every generation through processes like crossing over and independent assortment. This creates a ton of variation for selection to act upon.
So how do we know if a population isn't evolving? Is there like a baseline?
There is. It's a theoretical baseline called the Hardy-Weinberg equilibrium. It describes a hypothetical population that's not evolving.
What conditions need to be met for that?
5 main things. No mutations, mating must be random, no natural selection, the population has to be extremely large, and there can be no gene flow, no individuals entering or leaving.
Which basically never happens in reality, right?
Almost never, but it's incredibly useful because it's a benchmark. If a population's allele frequencies don't match the Hardy-Weinberg predictions, it tells us that evolution is occurring, and we can investigate which mechanism is driving it.
And it has practical uses too.
Yeah, like estimating carrier frequencies for genetic diseases. For PKU, phenylketonuria, which is recessive, if we know how many babies are born with it, we can use the Hardy-Weinberg equation to calculate the frequency of the disease allele and how many people are carriers, heterozygotes, who don't have the disease but carry the allele.
Okay, so populations almost always are evolving. What are the main drivers, the mechanisms causing those allele frequencies to change?
There are 3 main mechanisms. We've talked a lot about natural selection already. It's the only one that consistently leads to adaptive evolution, meaning organisms become better suited to their environment over time. It's driven by differences in relative fitness.
Which isn't just about being the strongest, right?
Not at all. It's
No.
about reproductive success, how many viable, fertile offspring an individual leaves compared to others in the population. An organism that survives for ages but leaves no offspring has 0 fitness in evolutionary terms.
And selection can push populations in different directions.
Yes, there are different modes. Directional selection favors one extreme, maybe larger body size, becomes advantageous. Disruptive selection favors both extremes over the middle. Imagine birds with very small beaks and very large beaks doing well, but medium beaks being inefficient.
And stabilizing selection.
That favors the intermediate types and acts against the extremes. Like hue and birth weight babies that are too small or too large have lower survival rates, so selection favors weights in the middle range. This actually reduces variation.
Does selection always reduce variation, though?
Not always. Balancing selection actually maintains variation. One way is through heterozygote advantage.
Like the sickle cell example.
Exactly. In regions with malaria, individuals heterozygous for the sickle cell allele, one normal, one sickle cell allele, are more resistant to malaria than either homozygote. The sickle cell allele is harmful when homozygous, but the advantage in heterozygotes keeps the allele present in the population.
Interesting. What about sexual selection? That seems like a special case of natural selection.
It is. It's selection specifically for mating success. This can lead to sexual dimorphism, those striking differences between males and females, think peacock's tails or lion's manes.
And that happens through males competing or females choosing.
Both. Intersexual selection is competition within one sex, usually males fighting for access to females. Intersexual selection is mate choice, usually females choosing males based on certain traits like elaborate songs or bright colors, which might indicate good health or good genes.
Okay, so natural selection is one big mechanism. What are the other two?
The second is genetic drift. This is all about chance events causing allele frequencies to fluctuate unpredictably from one generation to the next. It's purely random sampling, essentially.
And it matters more in small populations.
Much more. Imagine flipping a coin 10 times versus 1,000 times. You're much more likely to get a skewed result like 7 heads and 3 tails with only 10 flips just by chance. In a small population, random events can cause an allele to become much more or less common, or even disappear completely, regardless of whether it's beneficial or harmful.
Are there specific types of drift?
Two classic scenarios illustrate it well. The founder effect happens when a few individuals colonize a new location. The gene pool of this new founder population is likely different from the source population just by chance sampling.
Like if only red-flowered plants happen to colonize an island.
Exactly. The bottleneck effect occurs when a population size is drastically reduced by a sudden event, like a fire, flood, or hunting. The surviving population may have a very different allele frequency profile than the original population simply because of who happened to survive, leading to reduced genetic diversity.
Like the Florida panther example you mentioned earlier.
Yes, they went through a severe bottleneck, leaving them with very low genetic variation today, which can be problematic.
Okay, selection, drift. What's the third mechanism?
Gene flow. This is the movement of alleles between populations. Think migration individuals moving in or out, or even just pollen or seeds spreading between populations.
And this tends to make populations more similar genetically.
Generally, yes. It can counteract the effects of selection or drift that might be making populations diverge. It can also introduce new alleles into a population, sometimes beneficial, sometimes not, like insecticide resistance spreading among mosquito populations through gene flow.
So these three mechanisms, selection, drift, gene flow, are constantly shaping populations.
Oh.
But you said evolution isn't perfect, right? Why not?
Several reasons. First, selection can only act on existing variation. It can't conjure up the perfect allele on demand. It works with what's available.
Limited by history.
Exactly. Evolution is constrained by ancestry. It modifies existing structures. It doesn't engineer new ones from scratch. Like wings evolving from forelimbs, they're modifications of an existing plan.
Adaptations are often compromises too, you mentioned.
Right. A seal is great in water but clumsy on land. Our flexible hands are prone to sprains. Trade-offs are everywhere.
And chance plays a role.
Absolutely. Chance events, drift, selection, and the environment itself interact. An environmental change might make a previously neutral trait suddenly advantageous. It's a dynamic process, more like tinkering, as Francois Jacob famously said, than perfect engineering.
Okay, so these microevolutionary changes accumulate. How does that lead to the origin of whole new species? Darwin called it the mystery of mysteries.
Right, speciation. It's the bridge between microevolution and macroevolution. It's the process where one lineage splits into two or more distinct lineages. It's how we get the vast diversity of life.
How do we define a species, biologically speaking?
The most common definition is the biological species concept. It defines a species as a group of populations whose members can interbreed in nature and produce viable, fertile offspring, but cannot successfully interbreed with members of other such groups.
The key is reproductive isolation. Barriers that stop gene flow.
Exactly. These barriers prevent different species from mixing their gene pools. They can be prezygotic, meaning they act before fertilization.
Like different habitats, breeding seasons, courtship rituals.
Or even mechanical incompatibility, the parts just don't fit. Or gametic isolation, sperm can't fertilize the egg.
And if fertilization does happen,
Then postzygotic barriers come into play. These act after the hybrid zygote forms. Maybe the hybrid embryo doesn't develop, reduced hybrid viability.
or the hybrid is born but is sterile, like a mule from a horse and donkey.
That's reduced hybrid fertility, a classic example. Or sometimes the first-generation hybrids are fertile, but their offspring are feeble or sterile, hybrid breakdown.
So these barriers keep species distinct. But the biological species concept has limits.
It does. It doesn't work well for organisms that reproduce asexually or for fossils. And sometimes distinct species can hybridize, like grizzly bears and polar bears producing growler bears. Nature is messy.
How does speciation actually happen? geographically?
Geography plays a big role. Allopatric speciation, different country, is when a population gets geographically divided maybe by a mountain range rising, a river changing course, or colonization of an island.
The separated populations then evolved independently.
Right. They accumulate different mutations, experience different selective pressures, undergo different genetic drift. Eventually, they may become so different that even if they came back into contact, they couldn't interbreed anymore. Reproductive isolation has evolved as a byproduct of divergence.
Like those snapping shrimp separated by Panama.
A perfect example. Species on the Pacific side are most closely related to species on the Atlantic side sisters separated by the isthmus.
But can speciation happen without geographic separation?
Yes, that's sympatric speciation, same country. It's less common, but it happens. Gene flow is reduced by factors other than physical barriers.
How?
One major way, especially in plants, is polyploidy errors in cell division lead to extra sets of chromosomes. A polyploid individual might only be able to successfully mate with other polyploids, instantly creating reproductive isolation.
So a new species can arise in a single generation.
In plants, yes, quite often. Another way is habitat differentiation. If some individuals in a population start using a different resource or habitat within the same area,
Like those apple maggot flies. Some started laying eggs on apples instead of the native hawthorns.
exactly. This led to them breeding at different times and preferring mates from their own host plant, reducing gene flow even though they live side by side. Sexual selection can also drive sympatric speciation if, say, females strongly prefer males with certain colors or traits, leading to divergence.
What happens if these diverging populations come back into contact before reproductive isolation is complete?
You can get hybrid zones, areas where they meet and interbreed. The fate of these zones varies.
Hey, what are the possibilities?
If the hybrids are less fit than the parent species, natural selection can strengthen the prezygotic barriers, reducing hybridization. This is called reinforcement.
Makes sense. Less wasted effort on unfit offspring.
If the barriers are weak and hybrids are reasonably fit, so much gene flow might occur that the two species actually fuse back into one.
Or the hybrid zone might just persist.
Yes, that's stability. Hybrids keep being formed, maybe because parent species keep migrating into the zone. It shows that speciation is a dynamic process.
How fast does speciation happen? Is it always slow and gradual?
Not necessarily. The fossil record shows patterns of punctuated equilibria, long periods of little change, stasis, punctuated by brief periods of rapid change and speciation. But it can also be gradual. Speciation can occur rapidly, sometimes involving changes in just a few genes, perhaps affecting reproductive timing or pollinator choice in plants, or even a single gene controlling something like snail shell coiling direction, which mechanically prevents mating.
Okay, so from species, we move to the really big picture. Macroevolution. What does that encompass?
Macroevolution looks at the broad patterns of evolution above the species level. Think the origin of major new groups, mammals, flowering plants, birds. It deals with grand sweeps of history.
And mass extinctions seem to play a huge role in that history.
A profound role. There have been 5 major mass extinction events in the last half-billion years, where over 50% of marine species disappeared relatively quickly.
Like the Permian extinction,
Yeah.
the Great Dying.
The biggest one. Around 252 million years ago, maybe 96% of marine species, and many terrestrial ones went extinct, likely linked to massive volcanic activity in Siberia, causing extreme climate change.
And the Cretaceous extinction, 66 million years ago.
That's the one that wiped out the dinosaurs, except for birds, along with many other groups. The evidence strongly points to a massive asteroid impact near the Yucatan Peninsula in Mexico.
The consequences are devastating, obviously.
Immense, and it leads to a chilling thought. Scientists argue we're currently in a 6th mass extinction, driven by human activities like habitat destruction, climate change, and overexploitation. Extinction rates are estimated to be 100 to 1,000 times the normal background rate.
And recovery takes millions of years.
Typically 5 to 10 million years, sometimes much longer, for diversity to rebound. Mass extinctions don't just eliminate species, they fundamentally restructure ecosystems.
But extinctions also create opportunities, right? Leading to adaptive radiations.
Exactly. These are periods where surviving groups rapidly diversify, evolving new adaptations to fill the ecological niches opened up by the extinction.
Like mammals after the dinosaurs disappeared.
That's the classic example. Mammals existed alongside dinosaurs, but were mostly small and nocturnal. After the extinction, they rapidly diversified into the huge range of forms we see today. Radiations also happen when organisms colonize new environments with little competition, like the Hawaiian Islands, leading to unique species like the silver swords.
How did these major changes in form, these new body plans, actually arise? Is it just accumulating tiny changes?
Tiny changes matter, but major shifts can also result from changes in developmental genes, genes that control the rate, timing, and spatial pattern of development as an organism grows from an embryo.
So tweaking the instructions for building the body.
Precisely. Heterochrony, for example, is a change in the rate or timing of developmental events. Think about human versus chimpanzee skulls. Our skulls retain more juvenile proportions compared to chimps because jaw growth slows down relative to the rest of the skull during development.
Or the elongated finger bones in a bat's wing.
Another example of heterochrony. And sometimes sexually mature adults retain features that were juvenile in their ancestors. That's paedomorphosis, like some salamanders keeping their gills.
What about the spatial pattern, where things grow?
That's often controlled by homeotic genes, particularly Hox genes. These are like master switches that determine the identity of body segments, where legs go, where wings go. Small changes in these genes, or where they're expressed, can lead to major changes in body layout, like the difference between a six-legged insect and a many-legged crustacean.
And often it's not the gene itself changing, but how it's regulated.
Increasingly, we find that's key. Changes in the regulatory regions of DNA that control when and where a developmental gene is turned on or off can have huge effects without disrupting the gene's essential function. Like the loss of spines in stickleback fish, it wasn't a mutation in the spine-building gene, but a change in how that gene was expressed in the pelvic region.
It paints a picture of evolution as, well, not having a goal, not striving for perfection.
Absolutely not. It's crucial to remember evolution is not goal-oriented. It's constantly tinkering, modifying existing structures. Complex organs like the eye didn't appear overnight. They evolved incrementally from simpler light-sensitive spots, with each step providing some advantage.
Structures evolved for one thing can be co-opted for something else.
Yes, that's called exaptation. Think of feathers. They might have originally evolved for insulation or display, and only later became crucial for flight. Or the tiny bones in our middle ear, which transmit sound, they evolved from bones that were part of the jaw hinge in ancestral reptiles.
So even apparent trends, like horses getting bigger over time, aren't some predetermined path.
No, trends reflect the results of selection pressures in a particular environment over time. If the environment changes, the trend can slow, stop, or even reverse. It's an ongoing interaction, not a drive towards a specific endpoint.
So let's bring it all together. Darwin's mystery of mysteries, the incredible diversity, the underlying unity, the amazing adaptations of life, it all comes down to evolution.
Right. You start with genetic variation, the raw material for mutation and sexual reproduction. Then the mechanisms, natural selection, adapting populations to environments, genetic drift, introducing chance fluctuations, gene flow connecting populations, they alter allele frequencies over generations.
That's microevolution, and over longer time scales.
These changes accumulate, leading to reproductive isolation and the formation of new species, speciation.
And the grand patterns we see, the rise and fall of major groups, mass extinctions clearing the way for adaptive radiations, major shifts in body plans driven by developmental gene changes, that's macroevolution.
It's all interconnected. From a single DNA-based change to the sweep of life across continents and through millennia. Understanding evolution truly is the bedrock of all biology. It explains how organisms work, how ecosystems are built, how life responds constantly to a changing world.
Which really sets us up to understand everything else in biology we might explore.
Yeah.
Okay, here's something to leave you thinking about. We, humans, are now a major evolutionary force on this planet. Our actions driving climate change, altering habitats, using antibiotics and pesticides, are actively shaping the evolutionary future of countless species, very much including ourselves. The evolutionary changes happening now due to our impact will be studied by whatever intelligences exist millions of years from now. What kind of evolutionary legacy are we actually creating?
It's a profound question.
It really is. Well, thank you for joining us on this deep dive into the fascinating world of evolution. We hope this journey through Campbell Biology and Focus was valuable for you. This was the Last Minute Lecture Team.